and hosted by myself and my colleague John Lewick. For anyone new joining us today, each month we host speakers from around the world working at the forefront of GCE development and application, as well as closely related fields such as ribosome structure function, tRNA biology, therapeutics, directed evolution, and so on. And one of the things we really enjoy about this series is bringing together scientists from across different stages of their career. For example, today we'll hear from Professor Pezacki, who's been running an independent research program for almost 25 years, as well as Dr. Kat Vesely, who recently completed her PhD, is now beginning her postdoctoral work. And as always, if you are interested in presenting or you know someone who would be a great speaker, we would love to hear from you. All right, our first speaker today is Professor John Paul Pezacki, who is a full professor in the Department of Chemistry and Biomolecular Sciences at the University of Ottawa, where he also holds cross appointments in biochemistry, molecular biology, and immunology, and at the University of Ottawa Heart Institute. And he's also the founding director of the Center for Chemical and Synthetic Biology. John received his bachelor's in chemistry from the University of Toronto, followed by his PhD in chemistry from McMaster University, and then pursued postdoctoral training first at the University of Toronto and then at the Scripps Research Institute with Peter Schultz. John's research program aims to develop chemical biology tools to understand how viruses interact with and remodel host cells. His group works at the interface of chemistry and biology, building new methodologies to look at host pathogen interactions using a variety of approaches, ranging from activity-based protein profiling, RNA silencing, to bioorthogonal chemistry, and of course, genetic code expansion. And with GCE, among many applications, John's lab, they engineer nucleic acid binding proteins having new abilities, including cross-linking and chemical tagging chemistries to more effectively probe and look at RNA-protein interactions that are otherwise impossible to capture. Throughout his career, John's published 170 papers in top journals, including Nature Chemistry and Nature Chemical Biology. He's trained 35 graduate students, 19 postdoctoral fellows, and over 100 undergraduate students in his impressive career. So with that, John, we're very pleased to have you with us here today. Please do take it away.
Thanks so much, Rick. That was an amazing introduction and basically covered like the first 3 or 4 slides of my talk here as well. So that was really appreciated. Thank you so much.
You're
And
welcome.
thanks for the invitation. Today, I'm going to just give you a few snippets of our research that kind of touch on, what was just said about the applications of genetic code expansion in the context of RNA binding proteins. And of course, my lab works on RNA viruses, and so that it makes sense that RNA binding proteins are of general interest. I always start, all my seminars with acknowledging the people in the lab, because they are the folks that do all the work, and they are kind of the special people who will go on and carry this field forward. And today I'll be telling you mostly about the work of Aaron Lundergan and Matthew O'Neill, with that was enabled by a couple of chemists making some of the unnatural amino acids. So my lab, as was mentioned, my lab works on probing host pathogen interactions, from discovery to kind of applications, innovations in therapeutics, and even to some degree, we've gotten into vaccine design recently, using some of the key tools of chemical biology. So we do some things that are kind of more routine in terms of like profiling, but most of our approaches, make use of sort of modern aspects of chemical biology, and the specific tools that we develop in our lab. So, the way my lab works is that we have, this side that's tool building. We do bioorthogonal chemistry. We have some reactions that we're quite well known for, like the SPANC reaction with nitrones and cyclooctines. We make other types of probes. Be happy to tell you about those if you invite me to your department for a full seminar. Today we're going to talk about genetic code expansion and how some applications of genetic code expansion can be used to make new discoveries in host virus interactions. Of course, this all kind of started with the in the Schultz lab, and I want to just tip my hat to some of my friends who were in the lab at the time developing some of the OG technology, including Lei Wang and Ryan Mell and Jason Chin, who are like good friends of mine at the time, and it's been wonderful watching them, emerge as leaders internationally in these fields. So, yeah, none of this would be possible without their work. And of course, I don't need to explain this slide to this audience. But for specific applications, I'll tell you kind of three minuets of that kind of illustrate how we are using genetic code expansion. So this first example is a recent body of work that was kind of just recently completed by Erin Lundergan, where she was exploring the use of genetic code expansion to do protein interaction mapping. And this particular protein is ORF3A. It's a membrane protein from the SARS-CoV-2 genome, and it has been proposed in the literature to be um, um, a protein that's involved in entry, but also it has been implicated in other aspects of the viral life cycle. So it could be a viroporin, but it seems to have other functions. And so we wanted to explore whether genetic code expansion could help us to identify some of those interactions through cross-linking. And rather than doing ubiquitous cross-linking, we have the opportunity with genetic code expansion to do site-specific cross-linking, which allows us to kind of map domains and those specific domains interactions with other biomolecules. And we have other projects where we look at protein RNA interactions in a similar fashion, and we did just write a review for Angavante on that subject. All right, so as I mentioned, this interesting viroporin protein that seems to have other functions was the topic of this study. And in the literature, there were other kind of information that kind of hinted at the possibility that this protein may be involved in other kind of interactions with the host, including RNA processing, stress and apoptotic response, as well as innate immunity and innate immune evasion. So the main function that's kind of understood about it is that it's a viroporin, and so it kind of changes the acidity in the endosomal pathway and kind of causes the release of virus components during entry. But we wanted to explore these other aspects. And so we wanted to go beyond traditional kind of brutal 254 nanometer cross-linking and use genetic code expansion. So we used azidophenylalanine. There are other side chains that you can use that have diazarines. We chose to go with azidophenylalanine for this. The basic chemistry is kind of illustrated here. You lose nitrogen when you shine light on this azide, and it can form a nitrene that can react. The lifetime of the nitrene is short, and then it kind of undergoes a rearrangement to this dehydroazepine, which is also an electrophile. And so these kind of react with nucleophilic residues on other proteins and kind of trap the interactions. And we do this the way that everybody else does genetic code expansion. We have plasmids, the mammalian plasmids in this case, expressing tRNA synthetase, and our protein of interest with our amber suppressor codon and the site where that is non-membrane bound, so that we can capture the protein-protein interactions of the domain that I showed previously. Workflow is basically to express this protein and then look at the interactions with the host by UV photo cross-linking and doing interactome mapping by mass spectrometry, kind of similar to other interaction maps. And again, we avoid the transmembrane domains and we focus on the cytosolic domain of this protein and choose residues that are in flexible loops that may be easy to kind of access interacting proteins without disrupting the interactions. And you can see that we do get expression here with the blot. It's a lot less than wild type, but for a membrane protein, I was pretty impressed with Aaron to be able to get expression levels at that uh, at that level in mammalian cells, which was and enough for us to do mapping. And so we can do the pull down with the flag tag, my apologies, and we can see that we get other bands appearing in the pull down when we have AZF and 302 nanometer light. And these extra bands are representing the interacting proteins that are interacting with our ORF3A mutant. And we can then do mass spectrometry. And I'm not going to show you all the data, but just talk to you about the fact that actually those four categories that I told you about earlier are all kind of represented in the gene ontology analysis of the interactors. So kind of confirming that, in fact, these literature reports of this protein potentially being involved in multiple aspects of the viral life cycle seem to be holding true, and we get representative proteins that are in these bins. And here's some specific examples for those that might be interested. One of the ones that we saw that was related to immune response was STAT3, which is interesting, and Aaron was able to validate that that's a bona fide interaction that hadn't been reported before. And genetic code expansion helped us to identify that specific new interaction, which I think is powerful, application of this technology. All right, so we were able to do site-specific incorporation, photo activation of AZF to initiate cross-linking, and we reveal an interaction map and new interactions that had not been reported previously. And we feel that this approach allows us to detect more transient and weaker interactions with that site specificity and a higher efficiency overall of trapping with the use of genetic code expansion. So another protein that we're interested in from the SARS-CoV-2 genome is the NSP13. That's the helicase of the virus. It unwinds double-stranded RNA, enabling the polymerase to copy the genomic RNA and make progeny virus. It is a key kind of player in that process, and inhibitors of the helicase will basically block the virus from efficiently copying its genome, and this enzyme is nominally an antiviral target. So we use genetic code expansion on this protein to develop an assay by generating again azidophenylalanine, one of our favorite non-canonical amino acids. Because of course, we can do SPAC chemistry, strain-promoted azide cyclooctine chemistry with it, and incorporate new things site-specifically, in this case, fluorophore, to do FRET analysis. And we chose a number of different residues that are similar to azidophenylalanine, the aromatic side chains that, maybe, that are not likely to be structural or interfere with the function of the enzyme, because of course we want to report on enzyme RNA interactions with this. And we see that there are several mutants that express and click well, and others that like Y205 AZF that don't. And so this is kind of a representative of why we need to do multiple sites when we do these kinds of designs. But we do get good expression and we do get good clicking, and both of those things need to be optimized to get, to have this work, because, Y205 AZF expresses fine, but it doesn't undergo the click reaction very well, so which implies that it's maybe in a sterically hindered part of the protein. All right, so it's a simple FRET assay that allows us to measure binding. We have another assay that can measure unwinding, and we can do screens to look for inhibitors of this enzyme. We published the FRET assay because I think it's valuable, and it also illustrates genetic code expansion and its applications. We see a change in FRET due to the energy transfer between Cy5 and Cy3 and Cy5 that's directly attached to the protein through genetic code expansion. The other dye is on the RNA substrate, and we can measure interactions and inhibition of those interactions by inhibitors using this approach. And we have a whole story about that, where we did some AI-assisted docking, and we've got some really interesting inhibitors for this protein that have been validated with this and the traditional helicase assay. And this allows us to kind of distinguish between binding and unwinding. All right, finally, I'm going to end my talk telling you about some kind of protein engineering tool building that is associated with one of our longtime favorite proteins in the lab. This is the P19 viral suppressor of RNA silencing. We know RNA silencing is, being in the pathway that's involved in siRNA downregulation of messenger RNAs and also microRNAs and non-coding RNA regulation of mRNA translation. And this particular protein is a virus protein that evolved to combat the RNA silencing pathway in plant viruses. And it is a cool protein. I'll tell you some of the reasons why. It's very specific for the products of Dicer. And it has the specificity because it has 4 tryptophan residues that bind to let's see if this video will play. There we go. See the 4 tryptophan residues in red that pi stack and measure as a caliper these double-stranded RNA intermediates. Double-stranded DNA doesn't fit, only double-stranded RNA. And this kind of pi stacking interaction also makes this protein specific to binding to products of Dicer as opposed to single-stranded RNAs or longer double-stranded RNA sequences, because it needs to bind to both sides. And as it kind of does this caliper motion, it imparts a lot of backbone interactions with the phosphodiester backbone to lead to a very high affinity binding interaction. So this is actually picomolar affinity for the products of Dicer. And we've used this protein in various different assays that are represented here. And our most recent work so some of the work had involved genetic code expansion in the past, like these 2 papers, one reporting on the use of this protein as a delivery agent for therapeutic RNAs, and then another for looking at the interactions of the protein with different cellular RNAs using site-specific cross-linking. Our most recent work involved the use of bipyridyl alanine to impart catalysis to this otherwise RNA binding protein that is not an enzyme. And briefly, we used bipyridyl alanine to incorporate a Lewis acid catalyst to cause the cleavage of the phosphodiester backbone of the substrates, which could be siRNAs or microRNAs. We chose sites in the middle so that the product would spontaneously melt at room temperature, and we would not have no product inhibition. And because the protein only binds to double-stranded RNA, and we chose 2 residues, K67 and T111, to replace with bipyridyl alanine. But only one of these residues worked as an enzyme. The T111 site, K67 expressed and incorporated, but was not found to be a catalyst. So we had to validate that both of these enzymes incorporated and that they continue to bind their substrate. And these are EMSAs, basically showing that our protein binds RNA. and with nanomolar affinity, so we can retain that high affinity for the target. And now introducing copper to the bipyridal group, we can impart Lewis acidity and get this cleavage of the phosphodiester backbone. And what we find is that we did this cleavage assay in multiple ways. This is the way that I think shows catalysis the best. Here we have a, um, a quencher Psi3 probe that is kind of dark when double-stranded, and after cleavage, the release of the quencher gives us turn-on fluorescence. So fluorescence is a reporter of the enzyme activity nominally. And as we increase the substrate to concentrations that are well above the protein's concentration, we continue to see increases in product formation, indicating multiple turnover. We can calculate the Michaelis-Menten parameters for this enzyme, and the Kcat over Km is decent. In the range of like biological endonucleases. And not surprisingly, a good chunk of that comes from the binding, right? From the Km term, but Kcat is still pretty decent. And we can get complete cleavage of substrates in a matter of hours. And that's pretty good for kind of probing or kind of disrupting this pathway. And one of the kind of most interesting stories about non-coding RNAs in the recent past was the fact that hepatitis C virus, uses an endogenous microRNA to protect itself from degradation from host exonucleases cutting the RNA from the ends. It blocks the 5' end as kind of a pseudo cap through an interaction of microRNA 122, which is a highly abundant microRNA in the liver. And this was an interesting story that was developed by a company called Santeris that took this a microRNA inhibitor all the way to phase 3 clinical trials that they ended up getting bought by Roche. And the only reason why they stopped clinical trials was because there was an all-oral drug competitor that kind of won the race to curing Hep C. But these clinical inhibitors of microRNA 122 were curing patients up until phase 3 clinical trials. And our enzyme can basically destroy microRNA 122 at levels comparable to that inhibitor in cell culture models. And we did 2 of them, one subgenomic replicon and then one infectious model, showing that our bipyridal alanine containing P19 enzyme can basically cut and destroy the microRNA, giving a therapeutic response. So we kind of published this in Nature Communications a couple of years ago as the first unnatural enzyme that doesn't exist in nature that has endonuclease activity towards the small non-coding RNAs, the products of Dicer. And if you're interested in knowing more about that, you can read that paper. That brings me to the end of my presentation. Thank you for your attention. Thanks to all the great people in my lab once again. I'm happy to take questions.
That was really great. You've done a lot of really cool stuff. So I'll start off the question. Maybe if people have questions, they can go into the chat. If you're on the panel, you could raise your hand. But you've talked a lot about inserting non-canonical amino acids into your proteins. And I ask this of a lot of people. You show the stuff that works, obviously. But how much of the screening do you have to do in these like catalytic pockets or just in all for cross-linking to find the sites that work? Or are you just extraordinarily lucky when with doing?
Yeah, I think you have to kind of bias yourself towards luck. I don't kind of believe much in luck. I mean, you know, there's a few different things that obviously come into play, and I think the people on this call probably know better than I do. But we, certainly looking at secondary structure of the mRNA and making sure we're in kind of a good area, picking sites that are kind of similar to our non-canonical amino acid and finding sites that are not generally not, structural, are kind of key, right? And
Yep. And then do you
we've done this
screen a
enough
couple
times
sites?
now that we've had failures, but we've had successes, and we usually don't just do one, right? I mean, that's the kind of
Do a
also the
couple.
secret. You've got to do like make several mutants and test them, and then you go with the ones that work, right? And I think probably everybody does that, if whether they report it or not, right? So
Yeah. And then, so do you, when you're moving from like what is classically done in like E. coli and maybe even a little bit yeast, but when you're going into your mammalian cells, do you recode your DNAs for codon optimization to
for
improve?
sure, yeah, yeah, for sure, yeah,
That helps a lot. Yeah.
it does. Yeah, those are great questions. Absolutely. Codon optimization um, is a really important aspect for this as well.
I have one quick question. Maybe more in the details of your mass spec cross-linking work. Um, I think one of the challenges in the past has always been with cross-linking, you know, particularly with the phenylazide cross-linking is, the identity, the molecular identity of those cross-links, like you showed the chemistry, right? All the different forms of that can occur. And using mass spec to identify them has always been tricky. So when you do that mass spec work, are you do you are you only looking for cross-linked peptides that you consider as hits, or are you looking at all pep and basically anything that comes down, whether it's cross-linked or not? Like how are you sort of filtering through
Great
that
question,
data?
Rick. Yeah, no, I get where you're going with this for sure. And we have that same, I mean, we've been doing activity-based protein profiling for, 15 years now. And, I often tell people at conferences that we've done thousands and thousands of experiments and published, a few results, right? I mean, we've really spent a lot of time learning how to do these things right and,
Yeah.
to get good results. Uh, Obviously, it's like ideal if you can get the single peptide that comes from the modification, but that's not always easy. So for the work that I showed you here, we did not delve into the data that deeply. We really wanted to demonstrate just kind of the method and show that we're able to pick up these other proteins and kind of give them to the community and let say validate a few of them, but then just say, look, if you've got a particular interaction that you're looking for, and that we can talk and we can kind of propose maybe other ways to kind of probe it.
Yeah. Cool.
As any chemical biology technology, sometimes, you just think your tool is the answer to everything. And I think one has to be careful about that and really think about complementing the one method with, existing kind of traditional methods and kind of using a combination of approaches to kind of validate of new things that you might discover with your tools. I think
Yeah.
that's
Yeah.
that's what a
Can
lot of
you,
do.
yeah, for that, I mean, you use AZF a lot, and there's BPA or benzophenylene. Do you prefer AZF because it doesn't have this, it goes into destruction mode if it doesn't cross-link? Do you find, or do you use it for
I mean,
basically,
the diazurine does that too, right? I mean, you lose nitrogen and you get a carbene, and either it reacts with the protein or it reacts with water diffusion control, right? So
right?
both of those are similar that way. I would say maybe the diazurine is even more kind of destructive, right? Because I think the the rearranged alanine kind of structure of azidophenylalanine is maybe less electrophilic than an electrophilic carbene, right? So, um, yeah, so there's pluses and minuses, and yeah, I've thought about this a lot because I started as a physical organic chemist. I making carbenes and carbocations like a century ago, whatever. But these are really important kind of details. Um, so azidophenylalanine for us, so we have all the other expression plasmids for doing the diazurine, and there's the part of the problem with doing the diazurine is you have to make there's not a good commercial source of it that's like reasonably costed. So you have to make the amino acid, and that's a few steps. It's non-trivial. Shout out to my chemists, right? Alexa and Didier, who made, I think, $70,000 worth of bipyridyl alanine for that Nature Comms paper. If you buy bipyridyl alanine from Aldrich, you're going to pay, about $75,000 for the amount of bipyridyl alanine we needed for that study. So, the amino acid is always kind of a challenge. And also, we just found that azithromycin incorporates really well. Like
Yeah.
we are more lucky, like to John's point, we are more lucky with that than we are with other plasmids. So, why fight the bear? I mean, if we got one that works really well and we can do click chemistry with it, well, let's do that. So, I mean,
Yeah, this is important for,
that's kind of the
yeah,
mentality, really. It's maybe I'm oversimplifying it, and my
I
students
think that that's
won't be
right.
happy with that answer, but I do think part of it is, you go with what works, right? So,
Yeah, I think that people starting in with cross-linking, to tend to go with BPA or benzophenone first, right? And then
yeah.
they kind of run into a roadblock and they think it's impossible. So it's just, this kind of discussion I think is really good for the users that are getting into this to say like, oh, maybe I'm going to go with AZF first.
Yeah, and I can talk to you. I mean, certainly there's a physical organic chemistry explanation for why those two are quite different, right? And one is like a triplet state diradical of the ketone, which is reversible, and you can keep generating it. It probably lasts longer, so it diffuses farther, so you get more non-specific labeling. There's a deeper discussion that one can have around cross-linking that is going to be specific to your agent. So yeah, as
Yeah.
as with everything in science, there's a nuance once you dive scratch a
Yeah.
little bit down off the surface. So
Yeah.
And I agree with your sentiment about making your own luck. I wasn't trying to be condescending. Um,
Cool.